Okay. So good evening, everyone. Welcome to the live interaction session for week 8. My name is Ankita. I'm a PhD student at IIT Kanpur and I'm your TA for this course. Over the last 7 weeks, we have covered each of the topics for each of those weeks and have discussed some of the associated topics in that regard as well. So for today, we will have the last official interaction session for this course. And apart from that, there will be an additional session on Tuesday, which is 19th of March from 6 to 8 p.m., where we will recap the entire course in a somewhat brief manner. If there are any specific topics that are of interest to any of these students, we will focus on that. And other than that, any questions that I can find in that regard, I'll try to incorporate those as well. And any additional techniques or any information that is interesting or that might be relevant to anybody's individual careers, that also can be brought up and we'll try to address those as well. I will also recap the reference books that I have cited during this course with respect to whatever topics were discussed for that week. And with that in mind, we'll come to a completion for the entire course and you can prepare for your exams well in advance. So for today, we will follow a similar pattern as we have before as well. It will be a slightly shorter class than usual because most of the topics were elaborative have already been covered in one of the previous sessions, or some topics are very specific to the video lectures, for example, the one on stem cell editing, etc. So I will not be getting into those topics, but other than that, we will discuss the basic ideas that were a part of this week, which are different types of cells, the diversity that we see, how they are useful for scientists, how they're relevant to research, and what can be their potential applications when we want to make them useful for humans and for the population at large. So that will be the main focus of the discussion part. Apart from that, of course, we'll also go through the portion that is covered in the assignment for week 7, and also a few questions for sampling the idea of the topics that were given in week 8. And I will discuss the answers for, or at least related topics for week 8 as well in that extra session. And keeping all that in mind, we'll start the interaction session for today. If there are any questions, please do interrupt. I will try to address them in the best possible manner. So first of all, uh, just give me a second. First of all, I will start off with the I'll just reshare my screen again. Just give me a second. I'll start off with the assignment questions that were part of last week, where we looked at cell death, aging, apoptosis, the concepts of senescence, etc. So I think it should be visible now. Okay, so yeah, considering the topics that we had last week, where we looked at the differences between senescence, aging, the different types of cell death, whether it is programmed, whether it is necrotic, or what are the processes that are coming into play to make that possible, make the process of cell death possible, which was about the intrinsic and extrinsic pathway of apoptosis. We talked about the way in which it is regulated, what are the key messengers that are operating in order to facilitate cell death in that organized manner, and how it is different from necrosis. So these were the broad ideas that we majorly looked at. So the first question is about rapamycin. I think we had discussed this as well in the last interaction section. The drug rapamycin is an inhibitor of which of the following enzymes? So in the options, we have mTOR, AMP kinase, glucokinase, or none of the above. Now, the very name of this particular intermediate in the signaling pathway, which is mTOR kinase, is indicative of the function that it is associated with. So mTOR is the mammalian target of rapamycin. So this particular drug inhibits this kinase enzyme in mammals. It has a different function in bacteria. If it is something that is that interests you, you can certainly go ahead and look into it. But for mammals, the inhibitor rapamycin works on mTOR kinase. So that was the correct option. Then Question 2 was about cell swelling, membrane rupture, and inflammation. They can accompany which of the following, whether it is apoptosis, whether it is necrosis, whether it is senescence, or all of the above. Now, I hope that based on the last interaction session, the differences between these terminologies is clear to everybody. So just to briefly put it on, apoptosis is something that occurs in a very organized manner. It is otherwise understood as programmed cell death because the cell is keeping a lot of checkpoints in place and is carrying out the process of cell death in a manner which does not cause harm to the surroundings, which does not elicit any unwanted or unnecessary side effects upon cell death, and which is a necessary process also in a lot of contexts. For example, in development, it's a very desirable, very necessary process for development to occur in a proper manner. So that is apoptosis. On the other hand, necrosis is a more inflammatory form of cell death where there is a significant increase in not increase, but a significant swelling up of the cell, and which leads to a condition where the cell eventually ruptures and then it dies, and the cellular contents are available or not even available. They are sort of pushed out of the cell into the surroundings, and that can have a lot of unwanted, undesirable, and rather harmful effects. So that is the less organized or unorganized form of cell death. In contrast, senescence is a completely different idea. It's not a form of cell death. It is a sort of it's a sort of phase where the cell is in a inhibited state where it is not dividing, where it is quiescent, and it has attained that state after several rounds of division. Now, a number of factors could elicit senescence. If you remember, we had looked at hallmarks. So it one of the hallmarks, it was an hallmark of aging as well, and the context in which senescence is used is extremely different. So if we come back to the question, then the characteristic features which have been mentioned are cell swelling, membrane rupture, and inflammation. All of these are the characteristic identifiable features of a cell that is undergoing necrosis. So swelling up followed by rupture, and that eventually leads to inflammatory response being triggered. These are all components of necrotic cell death. Another phenotype I think I had also mentioned, or in case I haven't, another very obvious way or obvious thing that is observed during cell death is the formation of blebs. So bleb basically is the formation of these round structures around the cell membrane, which is characteristic of a cell which is about to die. So while normally you will, if you observe cells under a microscope, they will appear circular and very healthy. You will observe little blebbing around the edges, which shows that that cell is going to die. Now, blebbing is something that can also be observed in case of, in context of apoptosis, where the membrane is first blebbing and then is going into cell death. But sometimes in case of necrosis as well, it is observed. If you have to mention a characteristic feature, then membrane blebbing is usually identified in context of apoptosis. You can also look at the differences that we had mentioned between these two processes in the previous week's slides that I've already shared with you. So coming to question 3, which of the following is a pro-apoptotic gene? So we had seen that there is a family of proteins or pro-apoptotic and anti-apoptotic proteins that are classified, that have been identified, and that are well studied and well characterized into these two groups. And an imbalance of these factors, the pro-apoptotic and anti-apoptotic factors, is eventually what pushes the cell towards the process of death. So that is the reason why a cell is not continuously dying, because there are some external stimuli that are allowing that cell to survive, proliferate, and sustain itself. However, when these are overpowered by the pro-apoptotic factors that are in support of a cell undergoing death, then under such circumstances, the cell will eventually resort to programmed cell death or apoptosis. So the question is asking for one specific protein or one specific gene which is pro-apoptotic in nature. So among the options that are given, which talks about PCL2, MCL1, BAC as well as A1. So among them, BAC is the pro-apoptotic gene, while the others have differential response in context of apoptosis. So BAC is the correct answer for question 3. Coming to question 4, which of the following ions plays a crucial role in the control of various forms of cell death? That is necrosis. apoptosis, and autophagy. So when we talk about these three types of cell deaths, we have majorly focused on the protein, in particular the enzyme, that is very relevant in context of these processes. But if yeah, so but in context of I hope it is visible. Just give me a second. There's an issue with the window. Yeah, okay, should be fine now. Uh, when we talk about the ions that play a role in these different forms of cell death, then among them the most significant role is played by calcium ion Ca2 plus. If you remember that when the apoptotic pathway is triggered, then the release of cytochrome C is also accompanied by the release of calcium, and therefore it plays a crucial role in context of cell death. I think it was present in the diagrammatic representation that also we had seen in context of these different forms of cell death, and therefore that is the correct answer in this regard. Then we talk in question 5, we have been asked about PET scan. So PET scan is the positron emission tomography. It is a very commonly in the current scenario within the hospital environment. It is a test that is very commonly utilized. In particular, it is used for people, or it is prescribed or suggested for people who are either diagnosed with cancer, or to check whether they are at all, they have any metastases, or they have any sort of tumors that are forming within their body. In fact, it is also used for some other disorders as well, some neurological disorders in particular. And this technique is majorly focusing on the on tracking a radioactive tracer within the body. So that person is injected with a certain type of dye, which can be visualized within the scan. So it is easy to see where exactly that dye has traveled within the body. And it is found to be concentrated in regions which are very rich, or which have very high metabolic activity. Now, when we talk about cancer, then in case of cancer, these tumors, as we had, I think we had discussed in the previous session as well, that the amount of glycolysis which happens in context of tumors, particularly we discussed the Warburg effect, the amount of glycolysis that occurs can be easily seen, visualized, and can be concluded as an area which has a mass of tumor or a mass of neoplastic or cancerous cells within the body by this particular test. And in particular, it is even more relevant, that is during the follow-up process. So initially, after a patient has been diagnosed with cancer, and their prognostic approach has been, prognosis basically means how that patient will be treated. Diagnosis is the identification of a disease. So a patient shows some symptoms. Based on those symptoms, that patient is prescribed a number of tests, and taking the symptoms and the test results into consideration, the patient is diagnosed with a particular condition. So in this case, we are considering that to be cancer. After diagnosis has occurred, the doctor will prescribe a process or a regimen, a treatment regimen that they will follow, whether that could be chemotherapeutic, that could be immunotherapy, gene therapy, it could be radiation, surgery. It all depends upon what kind of a tumor that person is currently housing, or what kind of tumor is currently persistent within that individual. So when this occurs, and when a particular patient, suppose unfortunately, is in a very terminal stage of their cancer progression, then in such circumstances, usually the whole body PET scan imaging shows where exactly these metastases, which are the secondary tumors, they have formed. And when metastasis is very aggressive, we can see that it can be spread throughout the body, particularly concentrated in some regions like the lymph nodes, within the lung, within the bone. So bone metastasis is something that occurs very frequently, and that is usually not a very favorable circumstance. It is usually an unfortunate and a difficult to treat condition. Of course, there are certain cases where patients have recovered from it, but that is a very unfavorable scenario. Now, after this has been identified, when that patient undergoes the treatment regimen that is prescribed by the clinician or by the doctor, then at a certain regular intervals, these patients are again checked, or they undergo the PET scan again, and then a comparison is made between the initial PET scan that was present when that patient was untreated or treatment naive, that is the terminology that is used for such individuals, and what is the result of the treatment in these durations. So we expect to see an improvement, and that is monitored by tests like these. So this is the significance of using a test like PET scan imaging for particularly for cancers, but it is also used for other applications as well. So keeping all this background information in mind, PET scan uses one particular property of cancer cells that I just discussed, which is identified as the Warburg effect, and it highlights the importance of cancer cells and their utilization of glycolysis to proliferate rapidly and to generate the huge tumor cell mass, which will allow their survival. So for this purpose, they import very large quantities of glucose, and that is how you can detect where exactly these tumor metastases are localized within the body. So the correct answer would be option 1. Then in question 6, we were asked which of the following is not a hallmark for cancer cells. So if you remember, we had seen how Douglas Hanahan and Weinberg, they had identified certain numbers of hallmarks, and every 10 years or so, they provide more and more updates in this field of research. So there are some already established hallmarks, there are some enabling characteristics, there are some emerging hallmarks. So depending upon how research is evolving over this period of time, the concept of cancer hallmarks also evolves. So based on the options given here, we'll try to see which of them is not a hallmark for the for cancer cells. So resisting cell death is certainly a hallmark. That is, the cell will try to survive despite the ideal condition which would push that cell towards death. It will facilitate mechanisms which will not allow such cells to die. Second, there has to be replicative immortality, which means that they should not attain senescence. They should keep on dividing rapidly, abruptly, and should divide in a very abnormal manner, which will lead to mutations and a number of undesirable but cancer appropriate qualities. Genome instability is also a hallmark for cancer cells because that allows them to incorporate mutations which will allow cells to survive in a manner or allow them to survive under conditions which would ideally not be favorable. So because cancer cells are able to rapidly acquire mutations within them, therefore any population or any subpopulation within a tumor which carries a treatment resistant mutation will then take over and will survive by the basic principle of Charles Darwin, which says that there will always be survival of the fittest. So for example, if you are giving, say, a cisplatin drug, which we have discussed how it is it's a Taxol that is used, sorry, it's a platinum drug. Let's talk about Taxol, which is a microtubule targeting drug. So under such circumstances, if a cancer cell acquires a mutation which allows it to overcome the challenge of Taxol in the medium and allows it to survive under such conditions, then that cell will take over, and then that cell will proliferate and generate a treatment resistant or chemo resistant type of cancer population. And therefore, this ability of cells to incorporate or accommodate genomic instability is very, very crucial for their survival. So since all three options are correct, therefore none of the above would be the correct option for this question. Then in question 7, we were asked that a state of stable cell cycle arrest in response to diverse stresses or intracellular damage is termed as which of these following? So we just discussed this a while back, that there's a difference between apoptosis, necrosis, and senescence, and this particular definition where the cell enters a sort of arrested state where it is no longer responding to or is not entering a state of proliferation or is not responsive to the stimuli under all circumstances, then that cell is said to have attained senescence. So that is the correct option in this case. And in question 8, this actually is a question from the week before. So we had talked about the 3C technique at that point, I think in week 6. So the correct option for this, because it's something that we have already discussed, I'll get to the correct option directly. So the correct order in which you can perform the 3C technique or high C technique is cross-linking it, carrying out restriction digestion or chopping the chromatin into smaller parts or smaller fragments, followed by their ligation among themselves. So it will generate little fragments that are ligated towards the end. Then those fragments, they need to be purified. And finally, whatever fragments have been purified, they need to be sequenced by parent reading. And then that will give you the information regarding the genome and its organization. So taking this sequence in mind, the correct option is option 1, where we first cross-link, then we restriction digest, ligate, purify, and carry out paired end sequencing of the reads that have been obtained. Then in question 9, we were asked that the process of spreading of tumors from the site of origin to other sites within the body through either the lymphatic or the circulatory system is termed as which of the following? Benign, malignant, metastasis, or all of the above. So this entire process of the spread of a tumor from one location to another or to multiple sites within the body is known as metastasis. And what is formed as a result of this process, which are the somewhat smaller tumors that are localized in other regions within the body. Those are known as metastases. Just there is a difference in the spelling. That is META, meta, and then STASES instead of SIS, which is indicative of the process. These little structures are metastases, and often they are known as secondary metastases, and they can also be classified into micro and macro metastases based on what is their size and where they have localized, and their abundance as well. So this process would be called metastasis, and therefore option 3 is the correct answer for this question. And finally, in question 10, we were asked which of the following viruses can cause cancer. So there's papillomavirus, HPV, and EBV. The correct answer is all of the above. They are all oncogenic viridae or oncogenic viruses. So human papillomavirus, the hepatitis B virus, and Epstein-Barr virus, they are all oncogenic in nature. Therefore, the correct answer is including all of them. So with this, we've completed the assignment for the previous week. I hope it was clear. If there are any doubts, you can go ahead and ask me even at any later point during today's class. So we will now look at some of the topics for this week. Again, please interrupt me if there are any questions. Just give me a second. Okay, so the first, so basically the theme for this week was around understanding the diversity of cells. So the first not subtype, but the first type of cells that were extensively looked at were yeasts or Saccharomyces cerevisiae. They are unicellular eukaryotes. Very commonly, we know them as yeast, and however, the term yeast is a little bit of an umbrella term. There are different types of yeast as well. There is baker's yeast, there is brewer's yeast, and there is also fission yeast. All of these are different types of yeast. Among these, the particular model of interest that is mostly utilized is the Saccharomyces cerevisiae or S. cerevisiae. It is an extensively used model system even till date. Whenever some basic idea about eukaryotes is required. If some novel concept comes about, the initial testing is carried out in yeast, then we move on to Drosophila, then mice, then monkeys, and then finally we come to clinical trials in humans or research using humans. So it is an indispensable model system for studying eukaryotic conditions or eukaryotic contexts in biomedical research even till date. So keeping this in mind and highlighting how significant it is, we will try to understand what is the way in which yeast proliferates or yeast survive, or in other words, what does the life cycle of yeast look like. So as you've already learned in the course, there are two types or two genotypes of yeast that are identified. There is A and there's alpha. So when we talk about the A type of yeast cells or MAT A cells and the MAT alpha cells, they are different from each other, and they are capable of replicating only in a way such that the A and alpha can mate with each other. How does this happen? We'll come to the figure on the left. So what you see here is that the initial cycling that we see is a condition where the yeast cells are in a haploid state. which is indicated as 1N. So in the haploid state, we can see that the cell which is normally arrested in G1 will undergo cell division under circumstances which are conducive or which are helpful for the cell to divide. And under such circumstances, it undergoes mitosis, and the mitosis process gives rise to another haploid yeast. Then it goes through G2M and it again circulates back to G1. Now, under conditions where the yeast cell will enter into the sexual phase of its life cycle, then it will involve another type of or another subtype of cells, which are the mat alpha cells or the different subtype of cells. So in the sexual phase, what we see is that this G1 arrested cell enters a condition which is called as schmooing, or it forms this particular structure, sort of elongated, but not bud-like, but elongated pointy-like structure that you can see. And this structure is necessary in order to allow the exchange of genetic material between these two types of cells, which gives rise to the sexual nature of their fertilization or sexual nature of their reproduction. So these MAT alpha and MAT A cells form these smooth structures. They undergo exchange of genetic material, and when this is completed, then the cell that is the result of fertilization between these two can undergo sporulation, germination, and can again generate haploid cells in that circumstance. If that particular fertilized cell undergoes sporulation and germination, then it will generate more and more haploid cells, which will again enter into the haploid phase of the yeast life cycle. On the other hand, if the surrounding conditions are such that the cell is now entering instead into the diploid phase, then under such circumstances, we see that this particular fertilized cell will enter into the S phase. During the S phase, it will generate another bud. So yeast reproduction, while sexual in nature, the process is not the same as other mammals in general, but instead it undergoes the process known as budding. During budding, you can see that a tiny bud-like structure emerges. Then it goes through G2 phase, and finally in M phase, we see that there is division of the nuclear or genetic material as well. And as a result, the cell that will be formed will enter into a condition which is allowing it to again go another round of division, or and simultaneously it will release a bud. So basically, to sum it up, we see that the yeast can have three different stages or three different phases of its life cycle. We see a haploid phase, a sexual reproduction phase, and finally a diploid 2N phase. Each of these are interchangeable depending upon what are the external clues or what are the external stimuli or what is the external environment pushing that cell to do. So it can be context dependent, it can be situation dependent, and it will decide or determine how this cell is going to reproduce under those circumstances. So keeping this form of yeast life cycle in mind, we can now look at how exactly this process or this mating type at all is generated. So whenever we say mating type, it means there must be some genetic difference between these two. How does that come about? So what you see on the right is a representation of the way in which these genetic loci basically means a region of the chromosome. Locus is the location or region of the chromosome where it is present. So here we can see multiple genetic loci that are there. In particular, what is of interest to us are two regions which are at the extremes. There is HML on the left and HMR on the right. Both of these are heterochromatinized or silent region. When we say silent, it means that is a region which is not getting expressed. On the other hand, the region within the center, which is known as the mat locus, is which is for mating type or mating phenotype determinant. That is the region that gets transcribed and translated, and therefore it is expressed. It is at the center. Within the HML locus, we can see that there are two genes which are alpha 1 and alpha 2, which determine the alpha type of cell. And there is the HMR, which has A1, which determines the condition where it is a different condition. So what we see here, that the difference between a haploid A cell and a haploid alpha cell is that what is present at the center or what is present at the mat locus. If at the MAT locus the expressed gene is A1, then that particular cell will have MAT A phenotype or will be a MAT A type of cell. If it has the alpha genes at the MAT locus, then such a cell will be the haploid alpha cell, which is what is evident from the schema. I hope it is clear. Again, if there is any confusion, please interrupt me. I'll try to rectify it. So, oh, sorry. So these are the two subtypes that we see, which are capable of undergoing their independent haploid phases. Now, if there is schmoing and there is entry into the sexual phase, then these two can reproduce among each other. When they do so, what we see is the fertilized cell, or which is the diploid cell, that is said to have A alpha phenotype or A slash alpha, which is pronounced as A alpha condition. In the A alpha condition, we see that there are two copies of these genes, and while these two copies, they have the same HML and HMR region, the MAT locus basically has A1 and as well as it has alpha 1 and alpha 2 genes. As a result, this particular cell is capable of producing cells which can have either of these two genotypes. Now, how that will exactly happen is something we'll come to in the next slide. So what happens is that the yeast cell, since it carries both the HML and HMR regions having these individual genes, therefore, during its life cycle, one particular yeast cell is capable of changing the kind of genotype or MAT genotype that it carries. It can switch from A to alpha and from alpha to A. Again, this depends upon the external conditions, under certain stress conditions, under certain nutrient availability conditions, based on whatever is present. Just a second. Based on the stimulus that is present in the surrounding environment, yeast cells are capable of undergoing these changes or mating type switch from one genotype or one matched genotype to another. So how does this happen? This occurs by a process that we have already studied in one of the previous weeks, which is the process of recombination. So what you see here, start with the first figure on the left. So start with the figure on the left. What we see is the HMR locus and the MAT alpha locus. So among these, what you see is that initially this cell, because it has the alpha gene at the center or the alpha information at the center at the MAT locus, therefore the genotype of this cell or the mating type of this particular yeast cell would be of the MAT alpha type. It now wants to undergo or needs to undergo under certain circumstances, it needs to undergo the mating type switch into the MAT A type. So how does it do so? This involves the utilization of one particular very specific and very necessary enzyme, which is a restriction enzyme that is known as HO endonuclease. What does it do? We have discussed how endonucleases work. They cut within the DNA sequence at a particular region. And what this will do is that it will specifically make a cut or will cleave DNA at the MAT alpha locus after the alpha information has been completed. This 5' exonuclease is cutting at the MAT alpha nucleus, and then there is the digestion. If you remember, this was the exact way in which recombination in general occurs within cells as well. So after this cleavage has occurred, the other strand would be digested, and there is the involvement of RAT51, which forms that nucleoprotein filament, which will invade into the other strand. Which is the other strand? The other strand is the template, which has the HMR region, which carries the A information, what you can see in figure C. So once that happens, it invades into the other strand, and upon doing so, it will generate, it will find a region of homology, and upon finding that region, it will start to replicate. These are the, again, I'm repeating the same thing, but this is the exact way in which recombination in general occurs within the same, within the cell. So this is homology dependent recombination, or HR, as we commonly have known it. So after the replication fork has assembled, which is what we see in figure C, it will carry out or it will copy the sequence of the HMR template, and this template carries the A information, and therefore that will be incorporated into this particular strand. Once it has done so, we see that the region is extended as seen in figure D. This entire region has been duplicated or has been replicated, taking the template carrying HMRA region as the genetic information. And then there is displacement of the newly synthesized DNA, and the second strand synthesis is carried out, taking this as the template. Effectively, what you see at the end is that both the participants of homology recombination are both carrying the information for the A locus. And as a result, what we see is that the MAT A locus now carries the MAT locus carries the information for MAT A phenotype, in contrast to its initial condition where it had the MAT alpha gene. Therefore, this particular E cell has switched from the alpha to the A condition. Similarly, the opposite switching can also occur where an E cell which has the A genotype and phenotype will change into the MAT alpha condition. And in that circumstance, what will be different is that instead of HMR, the HML region, which is what we saw here, the HML region carrying the alpha 1 and alpha 2 genes will participate in the recombination process. And as a result, mating type will be switched into this particular one, which will generate a haploid alpha cell. So this is how the yeast mating type switch usually occurs. So this was about the way in which yeast cells normally survive. When we talk about yeast in context of research, particularly in biomedical research, we have just now said that they're very, very relevant for, in general, as a model for genetic studies, as an initial eukaryotic model for carrying out single cell isolated studies for understanding, for generating conditional mutants, for identifying the function of one particular protein which was previously unknown. It has innumerable applications. What we are particularly looking at in this context is the use of the yeast-2 hybrid system. It was also mentioned in the lectures, but I wanted to elaborate a little more on it and how relevant and how extremely brilliant this concept was. We want to get into that. So what is the need for these two hybrid systems? So I think we had discussed the ways in which DNA-protein interactions and protein-protein interactions can be studied inside the cell. So we had talked about the filter binding assay, we had talked about EMSA, and these are techniques that have evolved several decades ago. Now, when we talk about protein-protein interactions, well, there are more modern and more evolved techniques that are used in the current scenario. This particular bioinformatics, or rather molecular biology technique, uses the very simple principle of interaction between two proteins to identify their in vivo role, or their role inside the cellular system. So that involves two separate plasmids, which is what you see on the left, a bait plasmid and a prey plasmid. So the bait plasmid is one that carries a sequence of a protein that you will give as a bait, as the name indicates. And on the other plasmid is your prey, which will bind to the bait and will form and will interact with each other to form the two hybrid system that we are talking about. Let me read out for the legend for your clarity. So the principle of the yeast-2 hybrid system involves two plasmids, one which encodes for the protein X, which we are just naming here for sake of understanding, that is fused to the C terminus of a transcription factor DNA binding domain, and another plasmid which carries prey, that is protein Y, fused to an activation domain. Alternatively, the prey can also consist of proteins encoded by an expression library. So you can have an array of proteins which is from an expression library, and you can check which one of them do interact or do not, and therefore it can be a way of scanning for different interactions for the protein of interest or for the bait protein. So each of these plasmids are introduced into the yeast strains by co-transformation or sequential transformation or by yeast mating. These are ways in which you can incorporate both these plasmids into one particular yeast cells. Once that has been done, what we look at is that if the proteins X and protein Y, they physically interact with each other, then what that results in is that because they are tagged to another domain, which is in this context, it is the binding domain or it is the activation domain, it results in a complete system. So what that leads to is that it leads to transcription of a reporter gene. What you see here for ease of understanding, what you see here is a construct where there is a reporter gene. A reporter gene is something which, when expressed, can be detected by uh, a number of techniques. Either it can be a chemiluminescence assay, which means that there will be the emission of light. It can be the emission of color. It can be the emission of fluorescence, a number of different things. So this reporter gene is something which can report that there is an expression, that something has happened in the cell, as a result of which its expression is possible. There are many different types of reporter genes. For example, there is XGAL, there is luciferase. So these are all contextually used for different types of cells. When we see that the protein X and protein Y are interacting with each other, then we see that the DNA binding domain is able to bind to the UAS, or the upstream activating sequence, which is present much upstream of the reporter gene. And after connection between X and Y, or after interaction between X and Y, the activation domain is able to bind to the transcriptional machinery, which is assembled at the gene promoter, and it is activating the process of transcription, which is allowing the reporter gene to be expressed. So it is completing the circuitry that allows for the reporter gene expression to be initiated. Therefore, when these two proteins interact, we will see that reporter gene is produced. When they do not, you will not find any expression of the reporter gene. So this is a very easy way, and not easy way, but very accurate way of identifying whether these two proteins, or any two proteins within a particular cellular system, interact with each other or not. Now, the yeast to hybrid system has three minimal components that you need. The first would be an expression construct, which will synthesize your first protein of interest with the DNA binding domain. And this DNA binding domain will be able to bind to a nucleotide stretch that has defined specificity. That is what is termed as the bait, like we just discussed. The second one is an expression construct that is used to either express a second defined protein. Either it is one protein that is already known to be an interactor, or a protein that you are in that you speculate should bind to your protein of interest, and therefore you are specifically checking for the interaction between these two proteins. Or it can be part of a genomic library or expression library, like we just discussed, and that would be fused to a transcriptional activation domain, or AD. Frequently, it is termed as the prey. The third requisite for this particular assay is the reporter cassette, which has a DNA binding site where protein 1 will bind, and there will be it will also have a second region which binds to the transactivation domain, and therefore, in context of a minimal promoter, we will see that this reporter gene, which can be easily observed or easily scored, will start expressing, and therefore that can be detected. So what are the different possibilities that you might observe if you use such a yeast to hybrid system? This might be slightly hazy, but I hope it's visible. So what we see are the different conditions that can occur. The first condition, if we just introduce or incorporate the bait protein, which is the first plasmid X, into a cell, into an yeast cell, and you have a reporter gene which is bound to an auxotrophic, which is auxotrophic in nature, then you see that there is no growth, there is no expression. Okay, so the difference here in particular, I think I should have mentioned it beforehand only. What we were seeing in the previous case was a reporter gene, which was either giving us a visible readout or a visible condition, which is what you see in the second half. Here in particular, we are looking at auxotrophic reporters. Now, if you are not already familiar with it, auxotrophic promoters or auxotrophic genes in particular are those which are necessary for a cell to survive. And therefore, only when these genes are expressed, they are basically genes that have been mutated from the cell, and only when there is expression or external expression of these particular genes will the cell be able to survive, and we will observe growth. So they are survival essential genes, and these are cells which are deficient of those survival essential genes. So in the first case, when there is only the bait, we see that there is no expression of the auxotrophic reporter, and therefore we are not able to see any growth. In the second case, where we have a prey which is fused to an activation domain, we see that there is no interaction between the prey and the bait, and as a result, the transcriptional circuitry is not completed, and therefore the auxotrophic gene is not expressed, and no cell growth can be observed. So this helps us conclude that these two proteins that we were looking at are not interacting with each other. So bait and prey are not interacting. The third case where they do actually interact with each other, we see that there is expression of the auxotrophic gene, and therefore we observe that there is growth. You can see the strip at the bottom where there are these localized regions where growth has occurred. So this was the third set. So this is one way in which you can determine. You can use an auxotrophic gene as the reporter, and the other one, which is something that I've already mentioned, is use of a colorimetric reporter. If such a circumstance arises, then we see that when only bait is present, you can see white growth. When not in non-interacting bait and prey are present within a cell, then transcription is switched off, and therefore you do not see any color expression. However, when there is the cognate interaction between the prey and the bait protein, then the transcriptional circuitry is completed, transcription is switched on, and the colorimetric reporter genes are expressed, which gives us blue colored growth on the strip that you can observe below as well. So this is, as I have already mentioned, this is one of the most interesting and one of the most landmark techniques that are used for understanding the interaction of proteins inside a cell. You can obviously check it in a in vitro manner as well, but this is one of the best techniques that are used in this regard. So with this in mind, we will next move to the second cell system that was extensively studied. Now you're all mostly familiar with it. We have all studied these in our earlier classes about the plant cell. And in particular, the reason why we look at plant cells a little differently is because of the presence of some unique organelles that we do not find in case of other most of the other eukaryotes, and that is certainly the focus on the chloroplast. So if you look at the overall structure, you see the presence of the outermost layer, which is also the key differentiation point between plant and other animal cells, which is the presence of a cell wall. And what you see, let's just go over the labels one by one. We'll start with the innermost structure, which is, of course, the nucleus. Innermost as in the most significant region, which is the nucleus, shown in orange, and the different subregions within it, the nucleoplasm, nucleolus, nuclear envelope, and nuclear pore. Again, we have discussed each of these in extensive detail in the previous lectures. We have talked about the nuclear pore, the arrangement of nuclear basket, how it allows the shuttling of desirable cargo into and outside of the nucleus, the way in which the nuclear envelope is formed, what are the proteins that compose it, what are the diseases that can occur if the nuclear envelope assembly is not correct, which is majorly made up of lamins. That also is something we have looked at. The region specific expression of genes within the nucleoplasm or the nucleolus, and how that differentiation is being maintained. This composes the entire nucleus. Following that, we see the organelle which is closest to it, that is the endoplasmic reticulum. We have the rough and smooth endoplasmic reticulum, which is characterized by the presence of ribosomes. Some ribosomes we see are also present within the cytoplasm, not attached to the ER. Then we come to the next very relevant structure, which is the Golgi apparatus, which is crucial for the packaging and for trafficking of different proteins to the destined regions. And also it is very, very crucial for helping in protein folding in a proper manner. It allows for housing of histone chaperones, sorry, not histone chaperones, but protein chaperones in general, and heat shock proteins as well, which allow for the cell to combat the stressful condition and properly fold its protein, which will allow that protein to execute its function. Then we have the mitochondria, which is the main site of synthesis of energy within the cell. We have the peroxisomes, which have catalases and which are helping in managing the different types of species like the nitrous acids, or not nitrous acids, but the ions that might be generated, the radicals that might be generated within the cell under stressful conditions are taken care of by the peroxisomes. One very remarkable feature within plant cells is the presence of a huge vacuole. As you can see here, it's much larger than all of the other organelles combined. And therefore, the vacuole has a significant role in context of a plant cell. You might have come across it already. It helps in maintaining the structural integrity of the plant cell. It produces the turgor pressure, and it holds the cell shape in a manner which is necessary for sustaining the mechanical strength of a plant in the long run against all the different types of environmental conditions that it might have to face. Apart from that, structural stability is also being provided by the outermost structure, that is the cell wall. And that cell wall has certain pore-like regions, as you can see. And those pore-like regions are basically sites of accessibility between different cells. Why is that important? It's important because when we think about the way in which a plant survives, it is producing food or it is carrying out photosynthesis within the leaf, but the water that it needs, and that is being maintained by the phloem system within the cell, whereas the water that is taken up from the roots is transported throughout the xylem system. Despite that, there has to be another form of contact among these cells, and that form of contact is known as plasmodesmata, which is in this particular diagram observed as little pores within the cell. So that allows for intercellular connections. Now, keep in mind, these are not like pipes or open channels. There are also proteins which are present at the plasmodesmata that make sure that only the desirable things are being allowed to pass from one cell into another. And these are also significantly maintained, and they are necessary in order necessary for the cell to function in a proper manner. So with this, we have kind of looked at all of the different structures which are present in a plant cell. Now we talked about this production of energy or the production of food within the cell, within the plant cell in particular. So this is again another topic we are all very familiar with, which is photosynthesis. That is if we put it in layman's terms, it is the process of converting the energy from solar power or the solar energy into chemical energy, which means that you are harvesting the energy that is present in sunlight and you are converting it into a source of energy for the cell, and it is being done in a chemical manner so as to provide food for the cell, for providing energy for the cell, which will allow it to survive in the long term. Now, this was the broad idea that was present, and there is a genetic equation for photosynthesis also, where we see that there is glucose, water, and sunlight, which is ultimately converting or being converted into a condition where you have the presence of carbohydrates or the formation of carbohydrates and energy. Now, this was the basic idea that that we are familiar with from our childhoods. The question that now comes is that when we look at it in more detail, where exactly it is happening, then photosynthesis is being classified into two conditions: the dark phase and the light phase. The light phase, obviously, as the name indicates, is something that involves the investment of sunlight, and therefore it must occur during the day. And after this particular process has been completed during the night, we can see that, during the night, there is the second part, the dark phase or the Calvin cycle. So without getting into unnecessary detail, if we just look at this diagrammatic representation, we can see that there is the incidence of sunlight. When that is incident upon the cell, then in particular, the organelle which comes into play is the chloroplast. Here it is being schematically represented in green, and what you can see is a stack of thylakoids that are present within the chloroplast. So basically, if you section the chloroplast, you will be able to see little stacks, like coin-like stacks, which are present throughout. And the rest of the portion is the stroma. So this stack is made up of individual thylakoids. Within each thylakoid, photosynthesis is constantly taking place. So this is within these thylakoids, what we see is that this light is being taken up, and what is being produced as a result of it is ATP plus NADPH. Now these ATP plus NADPH are being utilized during the night for the Calvin cycle, which is a which is a cyclic process during which carbon is being generated, and as a result, we get ADP, PI, NADP, CO2, and the PGA3 as well, which is being released. And this generates the carbohydrates which are necessary for producing food and necessary for the sustenance of the plant at large. Again, during the next day, again, light is incident, and whatever is produced during the day is being utilized by the plant at night to produce carbohydrates. There is another class of so this Calvin cycle is otherwise also known as the C3 cycle because it involves 3 carbon compounds. But more evolved plants, like over the course of evolution, the newer plants, so to say, have evolved another form of processing carbons, which is known as the C4 cycle. And they both have their own advantages and disadvantages. It is something that has only evolved over time. So these are the various ways in which you can see that photosynthesis is also being evolutionarily monitored and optimized for plant survival. Next, we look at the growth of a plant. So it is not just the carbohydrate that is necessary. There are also plant hormones, which are very crucial. And these plant hormones are extensively utilized for our own needs as well, for our own research, for our own understanding of, or for even agricultural purposes, for expanding the culture of certain types of plants, for selecting for a desirable crop, so on and so forth. Multiple uses can be looked at. So when we talk about plant hormones, there are majorly like five groups or five major types of hormones that are observed. The first is auxin, which while it has multiple roles, it is majorly credited with the gravitropism of roots. Tropism basically means the tendency to localize or to move towards something. The word tropism by itself. When we say gravitropism, it basically means moving towards the earth or down into the earth. So that gravitropism is usually observed as a consequence of auxin. Then we have cytokinins. Now, cytokinins are majorly involved in cell division, and they help in expanding the cell or expanding or help in growth of the plant shoot in the shoot direction. This is in collaboration with another plant hormone group as well, which is gibberellic acid. And that gibberellic acid helps in cell division as well as in the control of stem elongation. Then we have the other two hormones. So auxin, cytokinin, and gibberellins, or gibberellic acid in particular, are the first three major groups of plant hormones that are extremely, extremely, extremely crucial. But we also have two other hormones, which are ethylene, and which is also known as forget something, auxin, cytokinin, abscisic acid. Yeah. So abscisic acid is majorly involved in the vasodilation, or to put it a little more simply, it is involved in maintaining the water content within the plant. So it helps in water stress. So depending upon the kind of environment that the plant is in and the availability of water and the requirement of water within the plant or the tree at large, abscisic acid has a huge role to play in that context. And finally, there is ethylene, which is involved in development of the abscission zone. It is extremely crucial for ripening of fruits, and it also might have some role in initiating the flowering of certain plants. So these are different hormones, and they are, as you can obviously see, they are all interacting with each other, and they all contribute to the growth and development of these plants. So keeping this in mind, the system that I've not elaborated on, but that was present as part of the video lectures, is one particular degron. Degron is a system that helps in degradation of a protein of interest. I have also given a schema on the right for your understanding. What you see is a degron that has a substrate. You can see that upon the binding of a substrate binding domain, there is it calls for the ubiquitination machinery, the E2 and E3 ubiquitin ligases, which come and which ubiquitinate the substrate that targets it for proteasomal degradation and removes that protein from the system. When this degron, as the name indicates, it is a degradation machinery. When this degron is tagged with some sort of an inducible promoter, then that helps us to um, control when exactly this degron would be operational, and therefore it will not be a permanently deleted protein or permanently degraded protein, but will be a context dependent situation. So for example, if I want this protein to be degraded on, say, the 10th day after a treatment with a fertilization, then I will be able to control that because I will introduce the inducer, which in this case is auxin, and the auxin inducible degron, otherwise known as AID, is extensively used in plant associated research. And that allows us to control that whenever auxin is applied to such a plant, then this plant will initiate the degradation of the auxin inducible protein of interest. So it is a degradation system which has been put into place in plants and is extremely useful for a large number of research applications. Now, when we talk about degradation systems, I thought I would introduce you to another concept. It's a relatively newer concept. Degrons have been known for a while, but more with time, what we see is that more targeted and more specific degradation is what people are looking at. So this concept of PROTACs is something that has come up in the past decade or so. And PROTACs basically are proteolysis targeting chimeras. The name PROTAC stands for that. What does that mean? When we say chimera, it basically means a fusion, that there is a genetic fusion that has formed the chimera that we are talking about. Now, what does this fusion involve? That is what you see in this diagram, that there is a fusion between two different um, protein moieties or protein domains, so to say. One, which is a ligand for a target protein, and the other, which is a ubiquitin ligase recognition domain. So what exactly happens? Suppose um, let's say we find one particular protein which is directly linked to a certain type of cancer. Say, for example, for breast cancer, one particular, the overexpression or the high expression of one particular gene is identified to be the reason why breast cancer patients are progressing so fast. And therefore, as a cancer research scientist and as a pharmaceutical company, I would like to deliver, or I would like to prepare and then deliver one particular drug that is able to degrade that protein, say protein X. Now for degradation of protein X, I want to generate a PROTAC. This PROTAC will have one region that is able to bind to protein X, has a linker region, and it has another region which is able to bring the degradation machinery or the ubiquitination machinery for the degradation to be carried out. So it is a small peptide. Basically, it's a small protein that you see which independently folds into these two domains, and it is linked to a linker region. So what this effectively is doing is that if you look at the schematic again, you are seeing that there is your target protein to which this particular ligand is binding, and it is also bringing the E3 ubiquitin ligase, for example, VHL, very close to that. When it does so, the ubiquitination machinery will identify protein X as something that needs to be targeted and needs to be degraded, and therefore it will start ubiquitination, which will be followed by polyubiquitination, and finally proteasomal degradation of this protein of interest. Once this is done, after that protein has been degraded, the PROTAC would be released and recycled, and then it can go and bind to other proteins. So what it is effectively doing is it is a very targeted degradation of one protein of interest, because you are using the ligand for exactly that protein. But the problem that is one of the preliminary problems that is identified with the use of PROTACs is the non-specificity. They have a very low half-life, and therefore their persistence or their availability within the cell is quite limited. So all of these like these shortcomings or these drawbacks are something that people are trying to overcome, and there have been modifications made for products. There are like, as you can understand in this case, if you have to have a ligand for a target protein, then that protein's structure and arrangement needs to be well known. So for proteins whose structure is not known, alternative strategies are being developed over time. There are bridged PROTACs. There are other different types of non-specific PROTACs which are all being looked into. So keeping this in mind, we will try to now shift our focus into the next region. But just till now, to sum up what we have talked about, we understood that plant cells are different in some ways. They have a different way of sustaining themselves than in our case. While the number of hormones in case of plant cells are limited, they're extremely crucial. They help in the both the sideways growth and for the length of the plant to be controlled. The existence of these hormones in the physiological condition within the plant system is something that we have understood and we have utilized for our own research purposes, which is the use of the auxin-inducible tachyon. And I also gave you one example of a different way of degrading proteins, which is via PROTACs. So keeping this in mind, we will next move on to the next type or the next diversity of cells, which are stem cells. Now, if you remember in the lecture, we had seen that stem cells are a subtype of cells which are capable of generating every other type of cells. They do not differentiate, they have the capacity for self-renewal, and they are extremely crucial for, their presence is extremely crucial within a tissue type in order to regenerate that particular tissue type under those conditions. So we talk about hair stem cells, we talk about skin stem cells, we have bone marrow cells, we have hematopoietic stem cells, all of these are present. There are only two locations within the body where they are not present. One is within the retina, and other would be something that you can look up in the lectures as well, that the absence of these make it very crucial for the individual, or for having strategies which can help in replenishing those stem cells. Why do we say that? Say, for example, like I just said, the two locations are the retina and of course the inner ear. So in case a patient or an individual suffers from an accident which leads to retinal damage or which leads to some sort of auditory damage, then in that case, those patients will, they either they lose their vision, or they lose their sight, or they lose their ability to hear. Therefore, having alternative strategies in place which can give rise or which can regenerate these particular cells and can restore the function or restore the abilities of those individuals are active areas of research in biomedical science. And they're very interesting. There are huge strides that have been made in this regard. And if it is interesting to you, you can certainly go ahead and read up on it. Now, coming back to stem cells, the topic of stem cells, let us first look at the timeline. So what are the ways in which they were at all discovered? So people had just seen cells dividing. Where at all did the concept of stem cell come from? So first we see in 1963, McCulloch and Till, they observed or they illustrated the presence of some self-renewing cells within the bone marrow of mice. Second, in 1978, hematopoietic stem cells were discovered in the human cord. When we say cord, it is basically the umbilical cord, and within the umbilical cord, we can see we have it's something we are already very familiar with. That in today's times, when babies are born, a lot of parents actually opt for the preservation of their stem cells because it can be extremely beneficial in the long run. Like I just said, that if an individual suffers these kind of damage, where there's a retina damage or there's an inner ear damage, then under these circumstances, that person's own stem cells can be utilized for regeneration and restoration of those functions. So these cord within the cord blood, hematopoietic stem cells can be observed. Then in 1981, mouse embryonic stem cells were derived from the inner cell mass by Martin Evans, Matthew Kaufman, and Gela Martin. Gail coined the term embryonic stem cell, ESC. Now, what is embryonic stem cell? If you see here on the right, you see that this is the embryo. Within the embryo, you have this sort of mass, the ICM or the inner cell mass of the trophoblast. These can be used for culturing, and this is what we understand to be ESC or embryonic stem cell culture. And what you see at the bottom is a electron micrograph of this exact same structure. You can see that there is a layer of cells on the outside, which is the trophoectoderm. The entire structure is the trophoblast. Within it, you can see this concentrated region, which is housing a lot of cells, and inside that is the inner cell mass. This is a blastocyst or a trophoblast, which has embryonic stem cells within the inner cell mass. Then in 1998, we see that James Thompson and his co-workers, they derived the first human embryonic stem cell line at University of Wisconsin-Madison. And then in 2006 came a remarkable progress in the field of stem cell research, where Shinya Yamanaka and Takahashi, they together generated the first concept of reprogrammable stem cells, which is where they induce the pluripotent stem cells by reprogramming adult mouse skin cells. Following that, they developed pluripotent stem cells from adult human cells, in particular from the fibroblast. So from an adult, you take a somatic cell, for example, a skin fibroblast, you de-differentiate it, or you take it to a primitive condition. Say, for example, if we have to draw a human parallel, for example, then you take an adult, you make that adult into a baby, and then you develop that baby into a whole other individual. So this is the way in which they carried out reprogramming of cells from an adult. They took skin fibroblasts, de-differentiated it into a stem cell-like phenotype, form the iPSC, or induced pluripotent stem cell. And from the iPSC, they generated a number of different cell types which were useful and could be utilized extensively in research and could solve a huge number of problems. So here we look at the different types of stem cells. First is the totipotent embryonic stem cell. Totipotent basically means that stem cell which can regenerate or which can differentiate into any cell type, including the extra embryonic layers, the endoderm, ectoderm, and mesoderm. They are able to do so. On the other hand, the next subtype, which is pluripotent stem cell, cannot generate the extra embryonic layers. So it can generate, while totipotent stem cells can generate the extra embryonic layers as well, pluripotent cannot. It can generate any of the other cells within the body, within the individual. Then come multipotent stem cells. So what you see is that during each step or during each gradation, the number of cell types that it can differentiate into is reducing, or it is becoming more and more specific, or uh, less and less differentiable. So multipotent stem cells are those which are capable of regenerating cells of similar lineage, not the same lineage, but similar lineage. So we can see that there are, for example, the prime example for this is the hematopoietic stem cell, which can generate any of the blood cells that are present within the system. So that is giving rise to the multipotent stem cells. So the clear difference between totipotent, pluripotent, and multipotent stem cells is something that you must keep very clearly in your mind. So how is it being used in context of research and for biomedical applications? So first you see here is a patient. From that patient, their adult cells are taken and it is being reprogrammed using some factors, reprogramming factors, otherwise known as Yamanaka factors, which are OCT4, SOX2, KLF4, and MYC. These four are being utilized to convert them into induced pluripotent stem cells. And then they are being modified or mutated or controlled in whatever manners are necessary in order to generate the gene of desirable mutation in the gene of interest or desirable conversion in the gene of interest, and then using definite specific factor or cell type specific factors, they can be differentiated into different cell types. So for example, you could have a primitive cell, you can differentiate into a neuron, you could differentiate into a cardiac cell, or it can remain as a stem cell, or it can differentiate into a prostate cell. So all of these can be different depending on what factors you are providing to that cell. So here we see examples of neural cells, cardiomyocytes, islet cells, and hematopoietic cells. Then after this has been done, after differentiation has been carried out, we can characterize what are the different types of disease phenotypes. We can study it within the lab. We can identify the disease specific molecular targets and use it for personalized medicine of the individual. So these were the far-reaching applications of iPSCs, but apart from that, stem cells in general have a huge role to play in one of the most tricky, but one of the most necessary processes or surgical procedures that are carried out, and that is for transplantation. So when we talk about transplantation, it depends upon the context. One form of transplantation that is quite commonly used in a certain types of cancers is bone marrow transplant, where from the marrow stem cells are isolated and they are introduced from a compatible partner. Bone marrow is harvested. One particular subtype of stem cells are isolated and they are introduced into the patient who is undergoing the transplant procedure. Some interruption. Yeah. So what we see here in the process is that first we have a donor from whom stem cells are collected, either from bone marrow or sometimes even from blood. Then this bone marrow or that peripheral blood is taken for processing, where stem cells are concentrated. They are more and more, they're increased more and more in number, and they're prepared for the process of freezing. And then they are subjected to cryopreservation. So the bone marrow or the blood can be preserved for an extended period of time to keep the stem cells alive until they need to be introduced into the patient. Then after the patient has undergone chemotherapy, that leads to non-specific killing of a large number of cells. then after chemotherapy has been carried out, these stem cells that were initially frozen can be introduced into the patient. Now, this was an allogenic donor transplant. When we say allogenic, it means that it is not from the same genetic background, or there is the exchange or transplantation being carried out between individuals from two different backgrounds. When an own individual's stem cells, or when an individual's own cells are being transplanted back into their own system after treatment, then such a form of transplant is known as the autogenic transplant. So this is autogenic when your own when your own cells are being introduced into you after some reprogramming has been carried out. When there is the transfer of a compatible partner's cells into your body, then that is an allogenic transfer. When it is from a different species into us, for example, when in certain cases some heart valves are replaced, when people have to undergo critical surgeries where they need a valve replacement, then in many cases bovine sources or pig sources or other different types of valves are often used, and they are often introduced into a patient or transplanted into a patient. Then that is a xenogenic transplant. Now, of course, the highest possibility of rejection of such an organ will be when the source is not the patient's own system, when it is external, and in particular when it is xenogenic in nature. So if it is possible, autogenic transplants are obviously most desirable, but if not, allogenic transplants are also extensively carried out. Apart from this, we can see that stem cells are also used in a huge number of other applications as well. For example, for understanding and trying to correct Alzheimer's disease, for baldness, for blindness, bone marrow transplantation, what we've already discussed, for deafness, diabetes, missing teeth, wound healing, muscular dystrophy, myocardial infarction, osteoarthritis, Parkinson's disease, spinal cord injury, and strokes. And also in a large cases for skin transplant, etc. as well, we can see that skin patches are introduced onto people in order to accelerate the healing process when they are burn victims, or when they have undergone some very serious trauma due to an accident or any other process of that sort. So with this in mind, we are completing the idea about stem cells as well, their use, their unique properties, their the ability to reprogram stem cells, their utility in research, their applications in context of patient treatment or patient therapy, and the extensive research which is being carried out for stem cells or in the stem cell research field is extensive. If anybody has interest in going into stem cell and regenerative research, you can explore. There are multiple options within the country and outside the country as well, and it's a very, very relevant field of research which will always continue to be relevant as well. So if it is something that interests you, please look into that, and of course it would certainly satisfy your scientific temperament as well. So with this in mind, we come to the final part of the discussion today, which is about the nerve cells. Now, what you see here is the structure of a neuron. And as you have already seen in the lectures, that neurons are not isolated cells. They are present with several other types of cells as well, which is what I'll come to next. But let's start off with neurons, the basic unit of the brain or the nervous system in general. So, of course, it has this peculiar and unique structure, where we see that this was the cell body from which are emitting these branch-like structures. Those are dendrites. And within the cell body, we see the presence of the nucleus as well. That is followed by a long stretch, which is called the axon. And that axon terminates with the sort of, again, uh, branched sort of ends, which are known as which is considered to be the axon terminal. And these are the regions where you can find the formation of synapses with the next neuron. So we see that the length of the axon is being coated by this blue colored sheath, which is known as the myelin sheath, which is also in very, very close association with another cell type known as oligodendrocyte. We'll come to that as well. So This is the basic structure of a neuron, which transmits information from one region to another. And there are different types of neurons as well. There are sensory neurons, which are able to receive information. There are motor neurons, which transmit that information. And then there are other neurons as well, which will carry out the execution of that particular initial stimulus. What you see on the left towards the lower end is a histological section of possibly some brain or brain tissue or nervous system tissue. And what you see at the center is the neuron, and you can see how extensively branched it is and how beautiful it is. It has these very thin extensions, which are these dendrites that are present, and you can see the presence of the relatively large nucleus as well. And it is very, very closely surrounded by a number of other very tiny dots, and those dots are indicative of other neuroglial cells, majorly astrocytes. And you can see how extensively enriched they are, and they are crucial for the sustenance of these neuronal cells as well. The neurons by themselves are a very small population, around 20-25% within the brain or within the nervous system, and they're surrounded by all of these supporting cells, which is what we'll come to next. So the different types of neuroglial cells and their functions are also well understood. There is, if we talk about the nervous system at large, there are two different types or two different subclasses. One is the central nervous system, the other is the peripheral nervous system. Within the CNS, we have different subtypes, which is astrocyte, ependymal cells, oligodendrocytes, and microglia. And you can see their different functions as well. For example, astrocytes, they maintain the BBB or the blood-brain barrier that controls that everything that is into circulation should not and will not be allowed to enter into the cell. And they form this sort of permeable barrier that only, not permeable, but sort of restrictive barrier, which controls what can or cannot enter into the central nervous system. Then come ependymal cells, which line the spinal cord and brain ventricles, and it produces the CSF or cerebrospinal fluid. Then comes the oligodendrocytes, which is what we saw in the neuron diagram as well, that they are responsible for myelination of the CNS neurons, and they also innervate those regions, and they provide the structural framework for the neuron axon as well. And finally, there is the microglia, which are the immune cells of the brain, and they are responsible for removing cell debris and cell pathogens by the process of phagocytosis. And finally, when we come to the PNS or the peripheral nervous system, then that is majorly composed of two other types. One is the satellite cell, and the second are Schwann cells. So satellite cells are surrounding neuron or the neuronal cell bodies within the ganglia, and they regulate the levels of neurotransmitters, whereas the Schwann cells are responsible for myelinating neurons in the peripheral nervous system. So what is being carried out by oligodendrocytes in the central nervous system is taken care of by the Schwann cells in the peripheral nervous systems. Additionally, they are also helpful for regeneration of neurons after injury. So within the peripheral nervous system, which is throughout your body, or any, like, your central nervous system is only your brain and spinal cord, right? The rest of your body is being innervated by the PNS or the peripheral nervous system. And therefore, if you suppose get injured anywhere else and there's a scab or there is a huge wound, then that region, while it initially hurts, there has to be recovery within that region, not only of the muscle, um, or the epidermis, which is the skin, but also of the innervation that was present in in that location, so that you do not lose sensation or do not lose innervation in that region, and that is taken care of by the Schwann cells. Okay, now we are coming to the last slide, which is for the way in which synaptic transmission is being carried out in neurons and how that is relevant to the action potential as well. So what you see on the left is the action potential, which is something I think you are probably familiar with, but an action potential is basically an imbalance or a shift that occurs within the maintained electrochemical gradient that is present inside the neuron. So basically what happens is that inside and outside of a cell, there is a difference in the electrochemical gradient. So there is a negative inside state that is maintained within the cell. Now, when there is a sudden shift in this electrochemical gradient or this voltage inside the cell, so to say, then that leads to generation of an action potential, because that is a signal. That is basically a the information being carried from one cell to another. And once this is carried out, it results in the release of certain components, particularly in case of neuronal cells, those are neurotransmitters, from the first presynaptic neuron into the next postsynaptic neuron. So we'll see the way in which it operates. So what you see in the first part of this figure is that there's a generation of an action potential at the nerve terminals. What it does is it releases or it opens up these calcium channels that are present. As a result of this, calcium is being taken up into the cell from the surrounding environment. What does it do? The entry of calcium allows the fusion of vesicles and release of neurotransmitters. So neurotransmitters are not freely circulated inside the cell. No, they are packaged into small vesicles, and then those vesicles are carried from the nucleus to the end. So imagine the length of an axon, which is the length of an entire neuronal cell. The nucleus is present at the cell body, which is at one end of the entire neuronal cell. And from the nucleus, after that protein or after the gene has been transcribed, gene for the neurotransmitter has been transcribed, it will undergo translation, and then from this point it needs to undergo packaging into a vesicle, and from there it needs to travel the entire length of the neuron, and then it has to assemble at the end towards the axon terminal. And once it does so, then the induction of the action potential will allow that vesicle to fuse with the membrane and to release whatever neurotransmitters it had inside. Upon release, it is being taken up by the next cell, which is the postsynaptic cell, and this is also controlled by the postsynaptic excitatory potential. So under those circumstances, their own ion channels open up or receptor channels open up, and that allows the entry of the neurotransmitter into the cell, and that carries the information from one to another cell. And all of this happens within a matter of not even a second, within a fraction of a second, and it allows the quick transmission of information from one neuron to another. So to sum it up, basically what you see here is a synapse. A synapse has two components. There is a presynaptic component, postsynaptic component. Synapse is the junction between two neurons. And therefore, you have the pre and postsynaptic neuron. Within these two, there is the exchange of information from one end to another. And that is controlled by the generation of an action potential. And this action potential is controlled by the shift or the flux of ions that occurs across the cell membrane. And once this happens, it allows for the transmission of neurotransmitter from one region into another. Neurotransmitters are nothing but chemical molecules because they are being circulated or transmitted by neurons. Therefore, they're specifically called as neurotransmitters. Some examples of this are something you're already familiar with. We have acetylcholine, we have serotonin, dopamine. All of these have different roles, very, very relevant roles. They can affect the way in which your brain functions, your mood, your ability to comprehend things, to understand things, and to respond in concordance with what is being told or instructed to the brain. So with this in mind, yeah, I think basically this covers up the topics I wanted to discuss for today. If there are any questions, please bring this up. And now I will share a few questions, sample questions. And once we do that, we will be done with today's class. Please let me know if there are any questions at any point. I hope I was clear today. I spoke a lot, but I hope it was somewhat understandable. If not, please mention it. I'll try to help you out. Yeah. Okay. So taking the topics that we have just discussed, we'll go through some of the sample questions from the previous year. So the first question is about stem cells, and which of these is the correct statement about stem cells? First, that they are self-renewing in nature. Second, that they can proliferate at a specific age bracket. They can give rise to two daughter cells that remain as stem cells, or none of the above. So let's go by process of elimination. One stem cell does not give rise to two daughter stem cells. It gives rise to one daughter stem cell and another, which will eventually go ahead and differentiate into the tissue type, which it is currently surrounded by, or whatever microenvironment it is surrounded by. Secondly, they do not proliferate within a specific age bracket. They have the ability to proliferate indefinitely, and that is unique and necessary for a cell to be considered to be a stem cell. They are indeed self-renewing cells, and therefore the first option would certainly be correct, and therefore that is the answer to this question. Secondly, which of the following can be used to arrest the MAT alpha type of Cerevisiae or Saccharomyces cerevisiae cells in a G1 phase-like state? Um, I don't think any of the options here. No. So this question is asking for which of the factors can arrest cells in the G1 phase: alpha factor, hydroxyurea, nocodazole, none of the above. None of these can do so, and therefore that is the correct option. Um, which of the following can correctly define an auxotrophic marker? So we talked about auxotrophs and in context of these two hybrid assay and how you can use an auxotrophic marker for studying and for monitoring the way in which the two genes may or may not interact with each other. So keeping that in mind, let us look at the options. The first is that genes that are necessary for cells to grow under an essential nutrient deprived condition, which is the correct option. Basically, auxotrophic markers are survival essential genes or nutritionally essential genes, which a cell absolutely needs for it to grow. Second, genes that are necessary for cells to grow in presence of drugs like hygromycin. That's not correct. Genes necessary for cells to grow under non-essential nutrient deprived condition. The name itself is contradicting the very purpose. So the correct answer for this is those genes that are necessary for cells to grow under an essential nutrient deprived condition. So option 1 would be correct. Then coming to the plastids. So the non-pigmented plastids are called. So we only talked about chloroplasts today, but there are different types of plastids within the cell. The option here are leucoplasts, chloroplasts, chromoplasts, and xyloplasts. Among these, leucoplasts are the colorless or non-pigmented plastids, whereas chloroplasts will have chlorophyll. Chromoplasts can either have red, orange, or yellow pigments, depending upon what context, what type of pigment they are carrying, and therefore their coloration is also changing accordingly. And therefore, the correct answer to this question are leucoplasts. In the budding yeast genome, which of the following regions have a heterochromatin-like organization? So we discussed the budding yeast genome in detail today. There are four options here: the RDNA locus, the centromere, the SMC gene locus, or the telomeres. So of course, the telomeres do have the heterochromatinized locus, but also the rDNA locus is also the correct answer to this question. And we saw the way in which those HML and HMR regions were silenced or were heterochromatinized, whereas the MAT locus, which is at the center, was something that was undergoing expression, and therefore during meeting type switch there had to be a crossover event that allowed the introduction of the other meeting type gene to be incorporated within this region for it to have the for it to be transcriptionally expressed or transcriptionally active. So the correct answer for this are both RDNA locus as well as the telomeres. Then in the next question, we've been asked that in the plant system, the coaxial membrane channel that links the cytoplasm, establishing cell-cell communication. This is something we also discussed, that this is a feature unique to plants and it is present within the outermost layer, within the cell wall, and that is known as the plasmodesmata. Therefore, these are cell-cell communication channels, and it is otherwise also understood to be a coaxial membrane channel, and it links the cytoplasm of one cell with the next one. Uh, in the next question, we have been asked that the ability of cells, which has an ability, or a cell which has the ability to produce a complete organism is termed as which of the following: totipotency, pluripotency, multipotency, or unipotency. So Obviously, multipotent and unipotent are not cell types that can do so. Among totipotent and pluripotent, what you need to keep in mind is that if you need a complete organism, then just forming the embryonic tissues or embryonic layers is not enough. The extra embryonic layers are also required, and therefore only a totipotent cell is capable of doing that. So the correct answer to this is totipotency, which is option 1. Uh, then in the next question, we have been asked that a fibroblast can be reprogrammed to cells similar to ES cells or embryonic stem cells by the co-expression of which of the following transcription factors. So we have already discussed this, that this is the concept of iPSCs or induced pluripotent stem cells. And the different transcription factors that we talked about were OCT4, SOX2, KLF4, and MYC, which are all of these options that are mentioned here. And therefore, that is the correct option, all of the above. Um, then we've been asked that which of these neurons transmit information between different parts of the central nervous system. So we talked about the sensory neurons, which are sensing information. Then the second types are motor neurons, which are transmitting information from one, not transmitting, they're executing the response to that stimulus. And there are neurons in the middle, which are interneurons that are conveying the information that was sensed and then relaying it to the motor neurons, which can carry out that activity. Take a simple example. For example, the reflex action is something that actually takes into consideration all these three subtypes or subclasses of neurons. So the first set, which is the sensory neuron, will sense that there is, for example, an open flame that you are touching. For example, the neurons in your finger, they have sensory neurons and they're sensing that there is some danger there or something harmful there. Following that, your motor neurons, which are present in the muscles of your hand, will carry out will allow or will force that individual to move their hand away from that open flame so that the individual does not get harmed, because that is the survival instinct. Now, this information is being relayed from the source, which is the open flame, to the brain, which responds which responds to the fact that now this hand muscle needs to be moved away from the open flame, relays that information back to the hand. So there is an entire system which is taking place, which allows us to communicate that this is the stimulus and therefore this should be the response. So between these three subtypes, the transmission of information is being carried out by interneurons. Then comes the last question, which is that a neurotransmitter that is released in the neuromuscular junction is which of the following? We have serotonin, or acetylcholine, dopamine, and glutamate. Among these, acetylcholine is released in the neuromuscular junction, and that is just a direct question that was there in the video lectures as well. So yeah, I think with this, we are coming to, yeah, we have come to an end for the topics I wanted to cover for today's class. Um, if anyone has any questions, please go ahead. If you do not have questions today, please assimilate whatever questions you have had throughout the course so that we can discuss it on 19th. Like I initially mentioned also during the session, that on 19th from 6 to 8, there will be an extra session. You can join at any point of time. You can come up with any questions that you might have had during the entire course tenure, any topic that was a little confusing or that you're not sure about or that you need help with in general. And we'll try to address that and clarify that as much as possible. We'll also try to go through some questions if that is feasible and anything that can be helpful for the exam as well. And I'll also discuss the topics that are part of week 8's assignments. Maybe not the questions directly, but I'll discuss the associated topics in that regard. So yeah, so if there are any questions, go ahead. Yeah, hi.
Ma'am, can you hear me?
Yeah, I can hear you.
Yeah. Ma'am, your classes were really good. Ma'am, actually, this week we have the exam, right? So, what would be the pattern of questions, ma'am? Only MCQ, or we have Phillips and other essay type
So
questions?
yeah, I'll just show it you. I think it is available on the website itself. I'll show it you. Just give me a moment. Yeah. So here you can see the exam format has been given in the announcement section on the website itself, on the portal itself. So you'll be able to see the type of exam that you're going to give, if it is a computer-based test or if it is a pen and paper test, and you can see whatever written questions are there.
Ma'am, your, I think, network is down.
Sorry, I got disconnected. Just a second.
Yes,
Um,
ma'am.
Yeah, so basically here it has been mentioned that questions can include MCQs, fill in the blanks, as well as essay type questions. So it will depend upon what type of test you've chosen, but yeah, majority of the questions are usually MCQ based only. There
Okay.
might be some questions which can give you a problem set that these are the conditions that you currently have. What would be your approach towards solving the same? So it can depend upon that. And if you want, you can try accessing some questions online. I don't think they're available because question papers are not usually there. But the format is similar to what you find in assignments. There will, of course, be some different questions. But yeah, that is the overall idea. And I hope you've all tried to complete the assignments to your best possible ability, because that actually adds to your overall score on the course. And based on that, you can be graded with, the gold level or the silver level, whatever it is, if you are in the top percentage of students who have qualified the course with the exam. And then that goes on the DEPTEL website. So try to perform the best of your abilities, and then you can add it to your CV. It's an achievement for you, and of course, it's a learning experience as well.
Okay. Ma'am, the text transcripts are not available, ma'am. Actually, it is there, but it's only the YouTube lectures transcripts which is there in the text transcripts, ma'am. Will that not be this PPT and all will not be available?
You mean for the video lectures by Professor Lalora?
Yes, ma'am. Yes, ma'am.
I don't think they're accessible right now. You can go back to the lectures whenever you need it. It's not available on the portal. Even I don't have it. You can find my slides accessible, but I don't think the PPTs have been made accessible this time. If you want, I can still check once if it is something that is required. But there is a book, a reference book, which has been uploaded on the portal that can be useful if somebody wants it.
Oh, okay, ma'am. Okay. So we can study from that, no?
Yeah, yeah, yeah. And you can go through any of the reference books that I had mentioned also, just whatever topic is relevant. Majorly, I had mentioned Cooper and Karp for cell biology. I
Okay.
had some epigenetic topics were from Turner, BM Turner. A lot of cell cycle topics were, not even a lot, the entire topic of cell cycle was from Alberts. It is one chapter, one big chapter, but it's beautifully explained. Yeah, these are the major books that you can look at, or any reference book, and of course internet for whatever understanding you want. And some specific topics, whatever extra information I've included, that has also been cited, so you'll get the reference in each slide. So from there also you can
Your slides
refer.
are there, ma'am, in the
Yeah,
portal.
they're
Okay.
uploaded. Yeah.
Okay, ma'am. Okay, ma'am. I'll refer that then. I think that
Yeah.
should be easy. Okay, ma'am. Thank you, ma'am.
Any other questions? Okay, if not, then thank you so much for joining. I hope the course was useful for you. It was a very diverse course with a lot of topic and a lot of information. But yeah, it should give you a very comprehensive view of cell biology as well as molecular biology at large. Again, let me stress on the fact that any doubts you have, please bring them in the last discussion session, because beyond that, we will not be able to have any discussion regarding this. If you want to, you can put any questions on the discussion forum. Somebody from the team will get back to you. But yeah, if any remaining questions are there, please bring it up on Tuesday and we'll try to solve them as much as we can. So thank you so much for joining. Have a good night, and I'll end the meeting now. So thank you.
Thank you.
Thank
Bye.
you, ma'am.
Thank you.